Hello everyone, and welcome to the second video that's part of the lectures from chapter 17. In this particular lecture, we are going to discuss the actual citric acid cycle. So before this, we talked about the pyruvate dehydrogenase complex, which prepares the products of glycolysis, which is pyruvate to acetyl-CoA to get it ready to enter the citric acid cycle. And now we'll talk about the actual what happens next. So overall, let's just talk about what the citric acid cycle is. It's also known as the Krebs cycle or the tricarboxylic acid cycle, the TCA cycle. So you may have heard it referred to as one of these other terms. It's referred, in particular, it's called the tricarboxylic acid cycle because one of the components of it during the citric acid cycle is a tricarboxylic acid, which we'll get to in a little while when we discuss the different steps. Overall, it catalyzes the balanced reaction where we have, we can think of starting with our acetyl-CoA that we made with our pyruvate dehydrogenase complex. This is going to eventually, we're going to eventually break that high energy thioester bond to yield just a regular CoA that's ready to accept some more carbons and be used again. The two carbons from the acetyl group are going to get fully oxidized into two carbon dioxide molecules. In addition, we're going to reduce some electron carriers. So we're going to have 3 NAD pluses and 1 FAD that are going to get reduced into 3 NADHs and 1 FADH2. Again, these reduced electron carriers are ultimately going to be able to enter the electron transport chain to participate in oxidative phosphorylation. And finally, we have one substrate level phosphorylation where GDP and an inorganic phosphate are converted to a GTP. And GTP, like ATP, can be considered a form of energy currency within the cell because it contains those high energy phosphate bonds that can be broken and used to help reactions go. So at this point during the citric acid cycle, once it's complete, we have the final oxidation of all the carbons from our fuel source, which in this class we'll be particularly discussing it as our fuel source as glucose. But as we saw in that diagram in the beginning of our other video, proteins and fats can also enter into various points of the citric acid cycle as pyruvate, as acetyl-CoA, and as a couple of other intermediates. Yeah, like I mentioned, this is where we generate reduced electron carriers and GTP that can help as essentially forms of energy or sources of energy. And truly, the citric acid cycle is really the central point of coordination for the metabolism of a whole lot of biomolecules. A lot of different biomolecules can feed in and out from different intermediates within the different citric acid cycle as part of the whole interconnection of metabolism as in both catabolism and anabolism. But as you know, here in this course, we're really focusing on glucose metabolism, and that's what we are thinking about here. Although in this case, the citric acid cycle is just the citric acid cycle. So let's get into it. So here is a diagram showing the complete citric acid cycle. It has 8 steps. Here we're going to show entering from the top with our acetyl coenzyme A that we just made out of our pyruvate dehydrogenase complex from pyruvate. And then let's look and identify some of the different components that we discussed from our overall complete reaction. So we're going to generate 2 carbon dioxide molecules. Those come from the 2 carbons that were in our acetyl group. We're also going to generate our electron carriers. There are 3 NADHs at various steps along the way, as well as 1 FADH2 that's generated. Down here, we also have the generation of 1 high-energy GTP molecule. And all of the other intermediates that you see here labeled in red are the different intermediate products that are formed along the way as part of the citric acid cycle that are relatively transient. And labeled in blue here are all of the enzymes that help to catalyze all of these different reactions. So, and I forgot to mention that of course, our coenzyme A is released after breaking that thioester bond. All right, so let's start going through each of the individual steps. So, like I mentioned, we start with our acetyl-CoA that we just made, starting with pyruvate as the source of this acetyl group. Acetyl-CoA has this really nice high-energy thioester bond, and breaking that bond is able to give this reaction an overall really large negative delta G. Acetyl-CoA is combined with oxaloacetate, so keep that in mind because we're going to eventually, at the end of this cycle, regenerate oxaloacetate to be used in another cycle. And what forms, when we combine these two together, is a citrate molecule. And citrate is a tricarboxylic acid. As you can see here, there are 3 carboxylic acids, hence the name. And because it's highly charged, it's relatively unstable, and so it's easy to decarboxylate and remove these carboxylic acids as carboxyl groups. And of course, because we broke that thioester bond in acetyl-CoA by transferring our acetyl group onto oxaloacetate, we have a free coenzyme A now. So that's the first step, which is catalyzed by citrate synthase. And as the name suggests, this enzyme helps to make citrate. Step 2 is catalyzed by an enzyme called aconitase. And this enzyme is named for actually this intermediate that forms, which we see here in the middle, called cis-aconitate. This overall reaction is a dehydration followed by a rehydration, which you can see here in this diagram. And so we start with citrate. Citrate overall is an achiral molecule. It has some symmetry that you can see here. The groups that are drawn on the top and the bottom are the same. But what happens here is the hydroxyl group and one of the hydrogens that are highlighted in red on the citrate are removed through a dehydration reaction to form a double bond. And then when this molecule, the intermediate, is rehydrated, the hydrogen and the hydroxyl group are replaced in these sort of opposite carbons from where they first started. And so the result of that reaction is a molecule called isocitrate. And because of the very particular shape of the active site in this enzyme, first of all, there are 2 new chiral centers where there were not chiral centers before. And like I said, the active site of the aconitase has a really specific shape to it, and so the reaction always catalyzed this particular stereoisomer of isocitrate. All right, so let's now talk about the next reaction. So the next reaction is catalyzed by isocitrate dehydrogenase. As the name suggests, this is a redox reaction, as you know from the dehydrogenase name of our enzyme, and the substrate that it uses is isocitrate, which was the product of our last reaction. So, so this is a redox reaction, so we are going to do a couple of things. We're going to reduce an electron carrier, so as you see, NAD+ is reduced to NADH. This is accompanied by an oxidation of our isocitrate molecule. You see this hydroxyl group highlighted in green is oxidized to a ketone. Also important to note is that we have our first decarboxylation right here. So we lose one of the carbons that was sort of part of our original acetyl group as a carbon dioxide. So overall, we've produced our first carbon dioxide and our first reduced electron carrier, which is NADH, as part of our whole citric acid cycle. The next reaction is catalyzed by an enzyme called alpha-ketoglutarate dehydrogenase. So alpha-ketoglutarate, which was the product of our last reaction. Again, we have a dehydrogenase going on, so we know we're having a redox reaction. So once again, we have our second reduction of our electron carrier, accompanied by an oxidation, as well as another loss of a carbon dioxide molecule. So overall, and something interesting that happens here as well is that there is another coenzyme A that now comes in, a reduced coenzyme A, that's going to form a bond with the substrate, ultimately making the product 6-enol-CoA. And so we once again, because we have this bond formed with CoA, that's over here, we have now this high energy thioester bond, which when we break it in a later step is going to have a large negative delta G. So the carbon dioxide that we produced is our final carbon dioxide. As you recall, we put in one acetyl group that has 2 carbons. Once we've made 2 carbon dioxides, that's all the carbons that we put in, we've now taken out. We formed our second NADH, but not the last. And we formed that high energy thioester bond. So that brings us to our 5th reaction. This 5th reaction is catalyzed by a 6-enol-CoA synthetase. And this is one of those enzymes that's actually named for the reverse reaction. So if you imagine this reaction proceeding from right to left, you'd have succinate being formed into succinyl-CoA, and you'd be hydrolyzing GTP to form GDP and an inorganic phosphate. But that's not what we have happening here. This reaction is going to proceed from left to right. So that's where this high energy thioester bond breakage comes in. Breaking this bond has a large negative delta G, and this is going to help with our substrate level phosphorylation, where GDP and an inorganic phosphate are combined together to form one GTP molecule. And this GTP molecule is the equivalent of one ATP. And this is the first and the only substrate level phosphorylation that we see during the citric acid cycle. So now that we formed succinate, we are ready for the sixth reaction in the citric acid cycle, which is catalyzed by succinate dehydrogenase. Once again, dehydrogenase, we know we're catalyzing a redox reaction. This reaction is going to use our other common electron carrier, which is FAD. And this is the only FAD that gets reduced to FADH2 during the citric acid cycle. Interestingly, this FAD is actually covalently linked to an enzyme, to the enzyme, succinate dehydrogenase, and it stays linked to the enzyme when it eventually gets used during the electron transport chain. Because as you remember, all of this is happening in the mitochondria, and that's also where the electron transport chain is happening, so the FADH2 can actually stay right where it is, and it gets used that way. And during the course of this reaction, we have the reduction of our FADH2, and so we have the oxidation of our succinate into a fumarate. And that brings us to our seventh reaction, which is catalyzed by fumarase. And so that double bond that we just made to make fumarate, we are now hydrating it to create a malate molecule. And this is actually going to create a chiral center right down here. And it gets ready for the next step, where we have another dehydrogenation, which brings us to our eighth step, which is malate dehydrogenase. This is, once again, dehydrogenase. We know it's a redox reaction. So this is finally our last NAD plus gets converted to NADH. That's a reduction, so now we're going to be oxidizing our malate substrate into our final product, which is oxaloacetate. And as you hopefully recall, oxaloacetate was part of the first step of our reaction, where it got combined with our acetyl group that came from acetyl-CoA. So it makes sense that the last step in this cycle would be to regenerate that first molecule that we started with. So overall, now we've made our third and final NADH, and we've regenerated our starting product, or we made the product, which is now going to be the substrate for our first reaction when we restart the cycle all over again. So now you might be wondering, you might be aware that the citric acid cycle only takes place under aerobic conditions. We also just talked about all the different steps and all the different reagents and components that go into the citric acid cycle, and none of them are oxygen. So why is it that citric acid cycle only occurs under aerobic condition? Well, the answer is that the electron transport chain, which we're going to be discussing in the next chapter, chapter 18, this process follows the citric acid cycle, and the electron transport chain does require oxygen as the final electron acceptor. And so when there isn't oxygen, the electron transport chain cannot proceed. And so all of those electron carriers that we produce during the citric acid cycle, which would normally get reoxidized during the electron transport chain, they can't if there's no oxygen. And so we essentially can't recycle those electron carriers. And so we end up building up high levels of NADH and depleting all of the NAD+. Also, all of the other products of the citric acid cycle will accumulate. And so as we've sort of learned about reaction kinetics, once the products accumulate, as well as you lack certain substrates and these oxidized electron carriers, which you need to carry out some of the reactions in the citric acid cycle, the cycle just can't go anymore. So it's not that the cycle itself necessarily requires oxygen, but without oxygen, all of the different components required to run the citric acid cycle go away, or they get built up and stopped up somewhere else. So at this point now you should be ready to answer the practice questions that are all about the citric acid cycle.